Hello, I'm Monish and this is the resistant modulatory efflux inhibitory effects of capsaicinoids. So that's quite a wordy title. So let's just hop into some key terms and ideas to understand how this study is working and what it does. So to start off, there's capsaicinoids, which are a family of compounds found in the genus Capsicum that are extremely pungent, and an example of this would be they're what make chilies spicy. So there's just some reported effects that capsaicin could be a good antibacterial, and a recent study dove into those properties and found that capsaicin and capsaicinoids have decent efflux pump inhibitory effects. So you may be wondering, what is an efflux pump? Well, an efflux pump is something that is a critical protein in pretty much all microorganisms. It's pretty much conserved as well as pretty the same across almost all microorganisms, so it's a very important thing. And it's used to pump out toxins, so it can get rid of antibiotics, which is the main problem here, especially with terms to superbugs, which are strains of bacteria that are especially resistant to general antibiotics or just normal thing you would do to kill it. And it's theorized that it's the efflux pump that is used to help get rid of the antibiotics so they don't be able to take an effect. So again, with superbugs, they are kind of a new prospect in medicine just because with the increased use of antibiotics, there's this higher chance of this bacteria just randomly surviving and then going to the next generation and it has better resistance against this antibiotic, and it goes on and on until there's some extreme resistance. An example being MRSA, methicillin resistant Staphylococcus aureus, which is probably the most famous superbug as of now. And it's crazy because it'll survive to a lot of normal antibiotics that you would think would kill it, and that is very difficult for normal doctors today because if superbugs are difficult to kill, they have a potentially high mortality rate that could be terrible in an outbreak, say in a densely populated city, and they could also confer their resistance across plasmids, so they could kind of propagate this ineffectiveness of any antibiotics, which is extremely dangerous if it gets into the public and say causes a pandemic or whatnot. So if you're able to use this efflux pump inhibitory effects, well, now you got something where you can reuse these antibiotics that would normally be not effective because they just be pumped out because they're blocked. That efflux pump gets blocked and the chain is broken, and now they have a possibility to actually have their effect. So going into the article, it was published in 2019 in the journal Bioorganic Chemistry, and they used 16 different compounds, but I'm going to focus in on two of them, which are capsaicin and dihydrocapsaicin. The only difference between them is that dihydrocapsaicin has a carbon double bond. It's just the hydrogenated form of normal capsaicin. And I chose these because they are the organic versions. Everything else was used from synthetic sources, and they were ordered from just chemical companies. And what they did was they determined a minimal inhibitory concentration, or MIC. Basically, that means like it's this concentration where it'll form a ring, so bacteria won't be able to grow. So that just kind of tells like how not to go overboard. So in this case, they operated on a concentration of 128 milligrams per liter. And what they did was they took it, they used ethidium bromide, which is able to be used as a marker because it's fluorescent, and then they put that in, they used the concentration of capsaicin, dihydrocapsaicin, different compounds, and they measured the fluorescence increase every 50 seconds over a long drawn out period of time. So they can see like how much ethidium bromide is tagged inside. So the more that's tagged inside, the less that's been pumped out. So you can see the more that's inside, the more efflux pump has been inhibited. So let's see what they got. This is the graph. It's been slightly edited to remove a couple of compounds just so we can focus on a couple of other, only the ones I want to. So 1 and 2 are the capsaicinoids, 1 being normal capsaicin, 2 being dihydrocapsaicin. And then Veripanel is just a standard drug used against Mycobacterium magnatus basically it's just like a standard control here. And then piperin is another plant-based compound. It's a comparison between plant-based compounds. And then ETBR is just the non-edited, only ethidium bromide tagged, just to see how it would be like that. And then another control being with blank. They just didn't put anything in there, just checked and stays about the same, which is expected. So an interpretation of this results, it's pretty promising, as the capsaicin and the dihydrocapsaicin, they were better, slightly better than verapamil, and they were significantly better than just nothing at all, or just the piperine. So that means they do have a definite market effect on how much is retained and how much the efflux pump is blocked. So that gives promising results, as this could be used in conjunction with other antibiotics to help fight these superbugs and kind of continue the biotic war that humans and bacteria have been going on with for millennia. So it's a promising prospect. It can help fight against this new rising threat in just the medical field. So that's the main thing here. This is the principal article I used with some extra articles, just finding some data on how they did stuff. And yeah, any questions?